This content is not designed for use as a medical reference to diagnose, treat, or prevent medical illnesses or trauma. If you have questions about the diagnosis, treatment, or prevention of a medical condition or illness, please consult your personal physician.
Hello, and welcome back. I hope you're having a great day. Today's lecture is a little bit different from the previous ones, as I'm going to be talking about 3 disorders instead of just one: Huntington's disease, amyotrophic lateral sclerosis, or ALS, and Creutzfeldt Jakob disease. I've lumped these 3 disorders together because although they are rare, I think they're important to understand and to be aware of. So let's get started by discussing Huntington's disease. Huntington's disease primarily affects movement, cognition, and mental abilities. The first case of this disorder was reported in 1872 by George Huntington. This disease is fatal. It causes the progressive breakdown of nerve cells in the brain. It's also 100% genetic, which is something we can rarely say about a disorder. If people have the gene for the disorder, they'll get it. Huntington's follows an autosomal dominant pattern, meaning children of parents who carry the gene have a 50% chance of receiving it and thus the disorder. Approximately 30,000 people in the United States have Huntington's disease. That's about 1 person for every 10,000 people. Most individuals show their first symptoms between the ages of 30 and 50. In 5 to 10% of cases, the first symptom shows up during childhood or adolescence. This is referred to as juvenile Huntington's disease. In a small number of cases, individuals have developed the disease in their 70s and 80s. Once someone receives a diagnosis, they typically have between 15 and 20 years to live. Much of what we know about the genetic basis for Huntington's disease is thanks to the work of Nancy Wexler, who has her PhD in clinical psychology. The disease runs in her family. Her mother died from it, and so did her grandfather and 3 of her uncles. She has devoted her life to studying Huntington's disease in the hopes of finding its genetic link. In 1979, Wexler and her colleagues started a massive research project in search of the gene that causes Huntington's disease. They went to Venezuela, where the largest known extended family with the disease was located. Here they developed a pedigree of over 18,000 individuals, a sort of genetic family tree, and collected more than 4,000 blood samples. Their data led to the discovery of the Huntington gene in 1993. From this, we learned that Huntington's disease is caused by a mutation in the ITI5 region of chromosome number 4. Chromosome 4 consists of what are called CAG trinucleotide repeats. CAG stands for the DNA building blocks of cytosine, adenine, and guanine. When these building blocks appear multiple times in a row, that segment is called a CAG repeat. A normal Huntington gene includes approximately 17 to 28 of these CAG repeats. A defective Huntington gene, which causes Huntington's disease, typically includes 40 or more of these CAG repeats, with some individuals having 100 or more repeats. The genetic test for Huntington's disease measures the number of CAG repeats in the person's gene. The larger the number of CAG repeats, the earlier the person tends to exhibit signs of the disorder. This means a person who is considered at risk can have a blood sample drawn to find out if they will, in fact, develop Huntington's disease at some point during their lifetimes. The blood test can't be used to determine when a person will show symptoms, only that they will show them at some point. Research suggests that 90% of at-risk people opt out of the blood test, so they choose not to know. Perhaps they worry about losing insurance coverage or the strain that this knowledge can put on interpersonal relationships, including with siblings who may receive a different genetic result. Nancy Wexler is an at-risk person, but she never took the genetic test that resulted from her own research. She always wondered if and when she would develop the deadly disease that plagued her family. In 2020, Nancy turned 75, and unfortunately, she started showing symptoms of the disorder. A diagnosis of Huntington's disease was confirmed through a physical exam by a neurologist. There's a wide variety of motor, cognitive, and mental health deficits associated with Huntington's disease, making it a form of dementia. We'll be talking about different types of dementia in upcoming lectures, but for now, let me give you a basic definition. Dementia is an overall term for diseases and conditions that are characterized by declines in memory, language, problem solving, and other skills that affect a person's ability to perform everyday functions. Some forms of dementia, like Alzheimer's disease, primarily affect cortical areas of the brain, such as the four lobes. These forms are referred to as cortical dementias. Other forms, like Huntington's disease, primarily affect the subcortical systems in the brain. These are referred to as subcortical dementias. The motor symptom most associated with Huntington's disease is called chorea. It is often described as twisting, writhing, undulating, grimacing movements of the face and body. Some liken it to resembling the movement of a puppet on strings. Other symptoms that fall under the category of movement include things like bradykinesia, or slowness of movement, rigidity, akinesia, or difficulty starting movements, dysarthria, which is an abnormal rate and rhythm of speech, and dysphagia, or problem swallowing food. Some of the cognitive symptoms include things like personality changes, impaired judgment, impulsivity, perseveration, inability to multitask, slow information processing, and trouble finding the right words for something. Finally, mental health deficits include things like depression, irritability, apathy, and mood swings. Neuropsychological testing can pick up many of these symptoms, as well as identify deficits not necessarily observed in everyday life. Neuropsychological deficits tend to fall into 6 categories. The first category is psychomotor slowing. Here, patients exhibit slowness processing information and in physical movement. They also exhibit slow or abnormal eye movements, which would make it difficult to accurately search for something in the visual environment. The second category is executive function skills. These include deficits in things like planning, organizing and sequencing, cognitive flexibility, and set shifting, which is your ability to shift attention between one task and another. Have you ever played the game Uno? If so, you know that you can play a card from your hand if you can match the top card on the discard pile in either number, color, or symbol. Thus, you have to be able to shift your attention between these possibilities as you're actively playing the game. This is an example of set shifting. The third category is memory skills. Here, patients exhibit deficits in the free recall of information. So, for example, repeating back a list of words that is read to them. However, they don't exhibit deficits on tests of recognition, like saying whether or not a word was on the list that was read to them. This demonstrates that patients can encode the information, they just have trouble retrieving it. They also show deficits in procedural memory, which, as we've talked about before, has to do with skill and habit learning, like learning how to ride a bike. The fourth category is emotion processing. Individuals exhibit difficulty in processing emotions, both those expressed visually, as when they look at a face, and those expressed through a person's voice. The fifth category is social cognition. Here, patients have difficulties with interpersonal relationships and in adapting to the needs of others. They also have problems behaving according to social conventions. And finally, the sixth category is visuospatial deficits. Patients have difficulty copying objects, mentally rotating objects, and perceptually integrating objects. For example, being able to see 5 large puzzle pieces and know that if you arrange them properly, they would form a face. Let's round up our discussion of this disorder with a look at the neuropathology behind it. So the basal ganglia system, the limbic system, and the frontal lobes are involved. Both the caudate nucleus and the putamen, which are part of the basal ganglia system, are atrophied in Huntington's disease. Since the basal ganglia system sends reciprocal projections to both the limbic system and the frontal lobes, we would expect to see deficits associated with these brain areas as well, which we do. You can also see general shrinking of the brain, with the frontal lobes themselves shrinking by as much as 20 to 30%, which could account for other deficits that are observed. Unfortunately, there's no cure for Huntington's disease. The best one can do is treat some of the individual symptoms. It's a devastating illness that gets passed down from one generation to the next. We'll shift our focus now to another rare but devastating disorder, amyotrophic lateral sclerosis, or ALS. This disorder was first reported in 1869 by a French neurologist by the name of Jean-Martin Charcot. But it really wasn't until 1939 that awareness of this disorder became widespread when it struck Lou Gehrig, one of the most beloved baseball players of all time. In fact, the disease is still often referred to by his name as Lou Gehrig's disease. And who can forget the ALS Ice Bucket Challenge in 2014, which was extremely successful in raising money for ALS research. ALS is a progressive, fatal, neurodegenerative disease that destroys motor neurons in the central nervous system. These neurons, which stimulate our muscles to contract, reach from the brain to the spinal cord and from the spinal cord to the muscles in the body. When these motor neurons die, so does the ability of a person to move voluntarily. People lose the ability to walk, talk, eat, and eventually breathe. In the later stages of the disease, they become completely paralyzed. ALS typically manifests itself between the ages of 40 and 70, but people can get it at any time during their lives. Average life expectancy is 2 to 5 years, and it's estimated that about 5,000 people are diagnosed with ALS each year. ALS is typically diagnosed by ruling out other causes, meaning it could take months or years to arrive at a diagnosis. In fact, average diagnosis time is 1 year. There is no cure for ALS, and currently there have only been 5 drugs approved by the FDA to treat some of its symptoms: Riluzole, Nuedexta, Radicava, Exservan, and Tiglutic. ALS typically has a gradual onset, with initial symptoms varying across people depending on which neurons are affected. Some initial symptoms may include trouble grasping a pen or lifting a cup, change in vocal pitch when speaking, progressive muscle weakness, tripping, dropping things, abnormal fatigue in the arms or legs, slurred speech, muscle cramps or twitches, cognitive and behavioral changes, and/or uncontrollable periods of laughing or crying. When symptoms start, 70% of people initially present with spinal changes, which obviously involve the spine, and 30% present with bulbar changes, which involve brain areas such as the cerebellum, medulla, and brainstem. Spinal changes affect lower motor neurons. Symptoms include a progressive, painless weakness that may originate in the arm or leg, and problems with speech or swallowing. Bulbar changes affect upper motor neurons. Symptoms include increased muscle tone, exaggerated deep tendon reflexes, and the presence of pathological reflexes like jerking of the jaw or the Babinski response in the foot. Researchers believe that individuals who start with bulbar symptoms generally have a faster decline, a survival rate of less than 2 years, and a significantly reduced quality of life. Things like sensation and bowel and bladder function are typically spared in both forms of ALS. We also know that both forms of ALS affect a variety of brain areas, including the frontal and temporal lobes, brainstem, and the basal ganglia, among other structures. Changes have been noted in both gray matter and white matter tracts. Some language areas, like Broca's and Wernicke's areas, are uniquely affected by the bulbar form of ALS. When I first learned about ALS in graduate school, I was told that it was a disease that caused a person's body to completely deteriorate while their mind stayed intact. I envisioned someone being trapped in their own body without the ability to communicate with others. Although some people do maintain normal cognition throughout the course of the disease, we now know that others do not. Approximately 30 to 50% of patients exhibit mild cognitive impairment, and about 10% go on to develop a type of cortical dementia known as FTD, which stands for frontotemporal dementia, which we'll actually talk more about in an upcoming lecture. I should note that the neuropathology we just discussed is more pronounced in patients who exhibit some type of cognitive impairment than in those who don't. And those who show signs of frontotemporal dementia exhibit the most neuropathology. In 2019, a review by Somia Benbrika and colleagues was published, breaking ALS-related impairments down into 3 categories: cognitive, emotional, and behavioral. If you were to administer a battery of neuropsychological assessments to ALS patients with cognitive impairments, you would most likely observe deficient performance on tests that examine executive functions, so working memory, set shifting, planning, reasoning, and so on. Verbal fluency, so the ability to freely recall verbal information from memory within particular categories. So, for example, recall every word you can think of that starts with the letter F. Social cognition, so how people process, store, and apply information about other people and social situations. And verbal memory, so learning and then recalling information, as with a grocery list. In regard to emotion deficits, patients with ALS have difficulty recognizing emotions that others are expressing, whether these emotions are portrayed in their faces or in their voices. They also have difficulty not only identifying the emotions, but also knowing whether the specific emotion is a positive or negative one. For example, they may not recognize the emotion sadness in someone and would not even be able to identify whether what they are seeing or hearing is something positive or something negative. Finally, behavioral deficits can include things like lack of initiation, lack of motivation, aggressiveness, obsessiveness, disinhibition, impulsivity, reduced concern for hygiene, and mental rigidity. Keep in mind that these are possible deficits that a patient might exhibit. Not all patients will exhibit all deficits, and patients will exhibit differing levels of impairment depending on their degree of underlying neuropathology. Obviously, these deficits will be the most pronounced in patients who develop frontotemporal dementia. As symptoms progress and the patient reaches the end stage of the disease, major complications tend to develop related to breathing, eating, and speaking. Over time, besides paralyzing voluntary muscle movement, ALS also paralyzes the muscles patients need to breathe. They might need to use a continuous positive airway pressure or CPAP machine to help them breathe at night. These are typically used by people who suffer from sleep apnea. Some people with advanced ALS may choose to have a tracheotomy, a surgically created hole at the front of the neck leading to the windpipe or trachea, for full-time use of a respirator that inflates and deflates their lungs. Unsurprisingly, the most common cause of death for people with ALS is respiratory failure. With regards to eating, people with ALS can develop malnutrition and dehydration issues because of damage to the muscles that control swallowing. They are also at a higher risk of getting food, liquids, or saliva down into their lungs, which can lead to pneumonia. Some people opt for a feeding tube, which can help reduce these risks and ensure proper hydration and nutrition. Finally, in regard to speech, problems may start off with a slight slurring of words, but eventually their speech might become difficult for others to understand. Many times, ALS patients may need to rely on communication technologies in order to interact with others. When it comes to what causes ALS, doctors divide cases into two categories: familial, meaning the cause can be linked back to family members, and sporadic, meaning the cause is not known. About 10% of ALS cases are familial and 90% are sporadic. Several genes have been found to be associated with both the familial and sporadic forms of the disease. Two major genetic contributors include the SOD1 gene, located on chromosome 21, identified in 1993, and the C9ORF72 gene, located on chromosome 9, identified in 2011. Since 1993, at least 50 other genes have been linked to cases of ALS. Harboring a defective gene alone, though, is not enough to trigger the disorder. Researchers believe that non-genetic risk factors play a substantial contribution to the etiology of ALS. Unfortunately, little progress has been made in terms of identifying and finding consistent scientific support for these factors. Two risk factors with consistent findings to support them are: one, being male. So men have a higher incidence rate of ALS than women, with a ratio of approximately 1.2 men for every woman. And two, smoking tobacco, especially among women after menopause. Other possible risk factors with less consistent evidence to support them include a lower body mass index, dietary factors, participation in high levels of vigorous physical activity, certain occupations such as welding, electrical work, and being in the military, exposure to metals like lead, manganese, and selenium, exposure to pesticides or viral infections, history of head trauma, metabolic disorders, and cancer. Yeah, that's a lot, isn't it? And chances are If these risk factors do end up showing a consistent link with the incidence of ALS, they are most likely additive. So for example, a patient might be someone who has a genetic risk factor, is a welder, and has a history of head trauma and cancer. Studying the non-genetic risk factors of ALS is extremely complex, and is most likely why we don't currently have any direct answers about their contribution. Okay, now let's turn to the third and final disorder we'll be talking about in this lecture, Creutzfeldt Jakob disease. Creutzfeldt Jakob disease, or CJD, is a very fast, fatal, and degenerative brain disorder. It's characterized as being a prion disease. Prions are abnormal forms of normal cellular proteins. The abnormal accumulation of these proteins results in the destruction of brain cells, which leads to the formation of lesions that resemble sponge-like holes in the brain. For this reason, CJD belongs to a group of disorders known as spongiform encephalopathies. CJD affects one person per million per population per year. So in the US, we're looking at approximately 350 new cases per year. So fortunately, it's a fairly rare disorder. There are 3 different forms of this disease: sporadic, genetic, and acquired. The sporadic form, which occurs for unknown reasons, accounts for about 85% of all cases and primarily affects individuals over the age of 60. The genetic form accounts for about 10 to 15% of all cases and is generally seen in midlife, so the 40s to the 60s. Researchers have found that a gene on chromosome 20 codes the biological blueprint for the prion protein. So far, over 50 different prion protein mutations have been identified. The final form, known as acquired CJD, accounts for less than 1% of all cases, and its age of onset depends on the age at which the individual is exposed to the contaminant and the incubation period of the disease. Acquired CJD is further broken down into 2 categories: iatrogenic and variant. The iatrogenic form is acquired through contamination through medical procedures, such as blood transfusions, corneal and meningeal transplants, and use of human-derived pituitary growth hormones. The variant form occurs from ingesting beef tainted by bovine spongiform encephalopathy, or mad cow disease. This variant was first noted in the UK in 1996 and is believed to have originated from sheep that had scrapie, which is also a prion disease. The infected sheep were turned into feed for cattle, thus infecting the cows that ate this feed and the people who would go on to eat beef from those cows. CJD is typically diagnosed through the use of EEGs, MRIs, and spinal fluid. As I mentioned earlier, it has a very fast progression. Most people die within 1 year of being diagnosed, with many slipping into a coma before death. Unfortunately, we currently have no effective treatment to either arrest or cure CJD. There are a lot of symptoms related to CJD, though not every person is going to show every one. There can be a lot of variability between people. As with Huntington's disease, all individuals with CJD eventually develop dementia affecting the subcortical systems in the brain. Among the symptoms they exhibit are movement deficits like sudden jerky movements, impaired balance and falling, tremors and muscle weakness, muscle stiffness, muscle twitches, difficulty speaking, and difficulty swallowing. We can also see frontal lobe deficits, including personality changes, memory loss, impaired thinking, disorientation, and problems with planning and judgment. Finally, some experience vision changes, such as double vision and insomnia, as well as a variety of mental changes, including anxiety, depression, agitation, apathy, mood swings, and hallucinations. Well, this was an uplifting lecture. Pretty horrifying disorders, though thankfully also fairly rare. In the next 2 lectures, we'll examine a more common disorder, Parkinson's disease. See you then.